All right. Good evening, everyone. Welcome to our April meeting of the Western Pennsylvania Mushroom Club. I'm the current president, Stephen Buckland, and I'm going to share some updates on the club with you all. If I can get it to go to the next slide. There we go. All right, so everyone should have received their March-April newsletter. Yeah, there's a lot of articles in there. And our next newsletter will be out on May 1st, and submissions are always welcome. If there's something you'd like to contribute to the newsletter, just reach out to our editor, Cecily Franklin. You can find the info for submissions on our website as well. In terms of walks since our last meeting, we've had two. First, there was March 26th at Hillman State Park. About a dozen folks joined us for a slightly snowy and cold walk out at Hillman to marvel at the diversity of lichens there. So this was our, I think maybe our first lichen focused walk as a club. And we found over 40 different species of lichenized fungi and about 15 different species of non-lichenized fungi. I just wanted to say thanks so much to Kara and Ben for your help organizing and leading that walk. I know I had a really great time and added many new species of lichens to my life list, so that was exciting. And then this past weekend on the 17th, I led two walks in Frick Park. The one in the morning, we found many different polypores, crusts, and rust fungi. We found this really cool Puccinia that was growing on a type of plant called rattlesnake root. So that was pretty exciting. I'd never seen that one before. And then in the afternoon walk, we continued to see polypores, crusts, and rusts. But we also took a visit to a patch of Gyromitra caroliniana, which is pictured here. That was really awesome. Thanks to Barbara for not picking all those mushrooms so that I could continue to take people to see them. Yeah, so that's what we've had in the past month. And we've got a lot of walks coming up. So these are all the club sponsored walks. The only one that's sold out here is the next one that's coming up, which is at Peters Lake Park with John Plischke III. It's a morel walk, and there was advanced registration. So if you didn't get tickets, you're out of luck. But there's plenty of other walks this spring. April 30th at Hillman, you can meet Kara Coulter at the,
This is the Stallion Trail, but I got to pinpoint it on the map for everyone.
Yeah, it'll be on the Stallion Trail. There's a big parking lot there on the map called the Old Airport Parking Lot. Carol will post directions about.
Well, that's not where I was going to go. Oh, maybe it is. I don't know. I don't think that's the parking lot though. It's on that same road that we were on.
Oh, okay. Cool. So Carol will post directions. Stay tuned for that. That'll be at 10 a.m. at Hillman on April 30th. Then the next day at Pine Ridge Park, you can meet Richard Jacob at the Pine Ridge Lodge at 10 a.m. for a morel walk. This will not be accompanied by a talk beforehand like it has in years past. I think our club often does a morel walk out at Pine Ridge, and usually there's a talk associated, but not this year. Then on May 7th, you can join club identifier Beth Dinger at the fountain outside of the Frick Environmental Center at 1 p.m. And then on June 11th, you can join club mycologist Kara Coulter at Beaver Meadows Recreation Area at 11 a.m. So those are all some upcoming walks, and I just wanted to take a second to remind folks that we request no one hunts a walk or foray location for at least 2 weeks prior to a walk or foray. And it's only through your cooperation that we can have successful walks. So yeah, we've got these walks planned. Hopefully there'll be more coming up. And since most of these don't have registration, hopefully folks that want to come can attend. All right. We've also got some walks in partnership with Allegheny Land Trust. A lot of these are sold out, but July 6th at Lynbrook Woodlands still has tickets available. And just some details on these. These are all organized and led by our secretary and identifier, Julie Travellini. Tickets are $5 a person. You can register at AlleghenyLandTrust.org/events, and it's limited to 20 people. No walk-ins are permitted this year. So get your tickets in advance, or just go on a walk there the day after if you didn't get tickets, but not in the 2 weeks before. All right. We've also got the City Nature Challenge coming up here at the end of the month. So the observation period is from April 29th through May 2nd, so Friday through Monday that weekend. And observations from Allegheny or any of the surrounding counties will count towards Pittsburgh's stats in this challenge. But there's a lot of other cities throughout the Northeast that participate: Columbus, Cleveland, Rochester, Ithaca, Philly, Baltimore. So I know we have members all over the place. If you're closer to one of those cities, you could go participate in one of those cities as well. But yeah, it's a great way to just get out, see what you can find, contribute some data to the iNaturalist database. We have 2 walks happening that weekend, but unfortunately the Pine Ridge Park Walk, that's in Indiana County, so that won't count as part of the City Nature Challenge. But you can join Kara at Hillman, and any organisms you take pictures of and upload to iNat will count towards Pittsburgh's stats. So, yay. Some upcoming meetings. So tonight we have, fingers crossed, our last virtual meeting of the year. We'll be resuming in-person meetings next month with our cultivation meeting. And these will be held at the Frick Environmental Center. We also are looking for volunteers for the cultivation meeting. So if you are able to arrive early, usually that meeting starts an hour early. At 6 o'clock, we begin the log drilling. So if you are available, you can contact Jared. He is our treasurer. So the best way to reach him is treasurer@wpamushroomclub.org. Is there anything else you wanted to share about volunteers, Jared?
No, that's it. Or you guys, I'll be on the meeting. You can message me personally within Zoom. Also, for those that want to bring a log, and if you're not familiar, if this is your first time, you're going to pick up a oyster mushroom kit. But also you can bring a log, and we will drill it, and then you can put shiitake spawn in it, and then you can wax it, and you'll have your log ready to go. Just as a little background on what logs work, any hardwood typically would suffice. Oak, maple are fine. You want to make sure they're probably around 3 feet or a little bit longer, so 36 inches to 40 inches long. And then in terms of how thick they are, probably about 4 to 8 inches in diameter, sort of like the perfect log. And it should be newly cut. And when I say newly cut, probably within the last few months. So let's just say 2022. We don't want logs that have been, cut, 2 or 3 years ago. Those won't work for our purposes. So if you have a log like that, feel free to bring that, and we'll certainly get you some spawn so you can plug that up. If you don't, that's fine. We have, as I said, oyster mushroom kits as well. And it'll be our first opportunity to be at the new Frick Environmental Center. So excited just to be at the new spot this year.
Hey.
There you go.
Thank you. All right. And now I've got some other events that are happening. The Tull Family Theater in Sewickley is going to be screening Fantastic Fungi again. So if you didn't get a chance to watch this, or if you didn't get a chance to see it on a big screen, you have another opportunity. So Friday, April 22nd through Thursday, April 28th. And you can use the code word mushroom at the box office to receive a discount. So head on out and go see a movie. Saturday, June 4th, there will be a fungus event at Powder Mill Nature Reserve. I actually don't have a lot of details on this, but Sarah's on here. Sarah, do you want to say anything about this event at Powder Mill? Sorry to put you on the spot.
That's okay. This is a family-friendly event. Drop in anytime between those times to come and learn about fungi. There'll be some lectures, some walks, and some hands-on activities.
Awesome. Yay. And if folks are looking for more details on that, what's the website they should go to?
Powdermill.org. I will put it in the chat.
Awesome. Thank you. Yeah, if you haven't been to Powder Mill Nature Reserve, there's amazing trails there, incredible biodiversity. I'm sure you'll be finding a lot of cool mushrooms in early June. And the same weekend is the FIPS BioBlitz. So that's an all-day event. The club will be leading a mushroom walk, I think at 11 a.m. I think I'm leading a lichen walk at 10 a.m. So if you wanted to go explore some urban lichens with me, feel free. I think Julie is leading a walk on something else, or is just going to be there tabling for ALT. So it'll be a fun day. Lots of learning.
I'm leading wildflowers at 1 p.m. Yeah.
Yay.
And I'll be floating back and forth between the ALT and Mushroom Club table.
Yeah. So the Mushroom Club will have a table. Last year, folks set up some microscopes and we're showing people spores and microscopic structures. It was pretty fun. Cool. So yeah, that's the first weekend in June, those two events. In the very near future, this weekend is our Mushroom Education Day. It is sold out. It will be at the Ohio Township Nature Center. Yeah, so the classes are sold out in the morning, but the afternoon walk is free and it's open to whoever shows up. So if you want to join us for a walk, you can meet us there at 1 p.m. and we'll hit the trails. But yeah, thanks so much to everyone who bought tickets. We're really excited to offer 3 classes in the morning. Here's a fun little flyer for the same event. So yeah, Julie is going to lead a class on using iNaturalist and how to participate in the City Nature Challenge. Then Kara is going to lead a class on morels. And then Espo is going to lead a cultivation class. I think folks are going to leave with some Stropharia grain spawn and a lion's mane grow kit. So yeah, super fun, awesome day planned this weekend. And then the last big event I have to share about is our Link Off Foray. This is the club's biggest event of the year. So this year it's going to be Saturday, September 24th, with a free walk in Cook Forest the Friday before. So yeah, we're really excited this year to be bringing some amazing speakers. We've got Arlene and Alan Bassett joining us this year. Alan is a distinguished emeritus professor of biology at Utica College of Syracuse University. He's a professional mycologist and has authored or co-authored more than 25 books. Alan is going to be helping us with mushroom identification, and he's going to be giving a table talk at the end of the day. Arlene will be one of our main speakers, and she has authored or co-authored 18 different mushroom books. And she's going to be presenting on polypores, which is the topic of her most recent book. So that's going to be really exciting. And they'll both be available for a meet and greet after their talks, or after the talks, so during the afternoon. And our other speaker is going to be Stephen Russell, and he is the author of The Essential Guide to Cultivating Mushrooms, and he's the founder of the Hoosier Mushroom Society. His topic is the next generation of DNA research. He recently sent us a synopsis of his talk, and I'm really excited to hear his presentation. So yeah, that should be a really awesome day at the Lodge in North Park. So we got the big space. Yeah. All right, our membership report as of yesterday, we're up to 602 members from 317 households. So thanks so much to all of you for supporting the club, whether this is your first year being a member or you've lost track of how many years you've been a member. We really appreciate it. And if you're watching the live stream of this or the recording and you aren't currently a member, here's some information on our dues and benefits. Aside from the benefits listed here, you'll also receive our bi-monthly newsletter, updates on walks and events. You'll receive a discount on admission to the Linkoff foray, and you'll get a mushroom grow kit if you're able to attend our cultivation meeting next month. So a lot of benefits. You should just join. All right. We don't have any mushroom ID awards to hand out tonight. Julie, did you want to say anything about the ID awards? You want me to just power through this?
I'll just say real quick, hopefully now that we're getting into the more active time, hopefully you all will get out and get your species and submit them preferably via iNaturalist to me so that I can go through and look to see what you got and give you your buttons. It's fun and it's free and you get a cool button.
Yeah, it is fun. I need to prepare my next list. I don't know, the 300.
Oh my gosh, that's going to take me forever.
Yeah. But yeah, if you haven't submitted a list yet, you don't have to start at 10. If you already know 25 species, you could start there. If you already know 50, you could start there. But yeah, it's just a fun way to keep track and see how many different species you know. All right, so next month's meeting, we already mentioned, is going to be our cultivation meeting at the Frick Environmental Center, and Jared shared some details on what to bring. So you must be a member to receive a free oyster mushroom grow kit, and you can also bring a log to inoculate with shiitake spawn. Logs must have been cut within the past 3 months or so, and yeah, a hardwood or broadleaf tree is going to be best. Oak, maple, those are ideal. And yeah, just a reminder, we are meeting at the Frick Environmental Center. So different location. Hopefully it will work out really well for us. Yeah, any questions before I introduce tonight's speaker? Let me go through the chat here. Oh yeah, I meant to include that webinar, June 8th. There's an Allegheny Land Trust webinar, Common Mushrooms of the Garden and Backyard. So yeah, if you want to learn more about what mushrooms are growing in your yard or garden, join that webinar on June 8th. You can register on the Allegheny Land Trust website. And yeah, I think folks clarified what kind of log. Awesome. Well, without further ado then, I will introduce tonight's speaker. So tonight we're really excited to welcome Dr. Matt Kasson to present Exploring Fungal Biodiversity and Ecology Across Central Appalachia. Dr. Kasson received his PhD in plant pathology from the Pennsylvania State University. from Penn State, where his research focused on using a native fungus as a biological control of the invasive tree species Ailanthus altissima, otherwise known as tree of heaven. He also has degrees from Paul Smith's College and the University of Maine. His current research areas include fungal arthropod interactions, biological control of invasive plants and pathogens, and the biology and ecology of historic and emerging diseases of forest trees. Dr. Kasson is currently the interim director of the International Culture Collection of Vesicular Arbuscular Mycorrhizal Fungi, and currently has research focused on the metabolites associated with interactions between arbuscular mycorrhizal fungi and their plant partners. He teaches undergraduate courses on general plant pathology and forest pest management, and offers special topics courses for graduate students, including advanced plant disease diagnostics. So yeah, I think at this point, I can stop sharing and turn things over to you, Matt.
Well, thanks, Stephen. I appreciate the invite, and I'm excited to be here today. I'm sad to report this is my first meeting, but not my last. So glad to finally be a member of the club, and hopefully in the next few years / decades / half century, I could devote some time to the club and my expertise, which probably lies a little bit outside what a lot of the other folks do, because they work on some weird fungi. But I'm really excited to be here today and talk about a number of projects that I've worked on and my students have worked on over the last 10 years or so. I started at WVU in 2014 as an assistant professor, and I'm now an associate professor of mycology and forest pathology. And as Stephen said, I'm director of INVAM, which is the world's largest collection of arbuscular mycorrhizal fungi, housed at WVU. So pretty exciting to have that collection here. Let's see if this will advance without a problem. Oh, it looks like it's a little wonky. Not sure why it's not loading. Let me see if I can go back. Okay, so of course, it's important that we acknowledge the students and postdocs and technicians that do a lot of the work that goes into this research. And I've kind of indicated my lab group here, current and past members, and kind of put green check marks next to each of those individuals that contributed in some way, shape, or form to the talk I'm going to give tonight, which covers quite a bit of diversity and quite a bit of projects of the last 8 years. I'm going to move this other screen because it's hard for me to see my titles. So I like to tell people, we're really good at finding things in the woods, but in particular, we're good at finding canker pathogens, which you can see here on the right. You can see a number of different cankers, whether they be target cankers caused by Neonectria or chestnut blight caused by Cryphonectria. Well, we study a number of arthropod fungal interactions, and that includes the spectrum from mutualism to parasitism. And we study a lot of specialized parasites that occur on all kinds of arthropods, not just insects, but millipedes and other arthropods as well, arachnids too. So I just wanted to start off by kind of showing a current zoomed out view of kingdom fungi. This was a paper published by Rokus's group in Vanderbilt just this last year in Current Biology, and kind of shows kind of the fungal tree of life in its current form. And I'll just point out that there's been a lot of changes in the taxonomy of the early diverging fungi. That's those outside of the dicaria, Zilpagomycota, Chytridiomycota, Blastiocladiomycota, Mucoromycota. A lot of these were formerly called zygomycetes and chytrids, and the names have changed, but their ecology and their roles in the environment have not. But we always have to keep up to date as professional mycologists who are publishing literature a lot. We have to keep up to date on how taxonomy is changing, how that affects how we frame things. My lab focuses broadly on fungal biodiversity discovery and analysis. And we look at fungus feeding millipedes, which I'm going to highlight today, canker pathogens of trees, which I'm going to highlight today, and some work we do on cicadas, which I'm going to highlight today. Yes, I'm going to highlight a lot of these things. I'm particularly specialized on the Entomopterales, these insect-destroying fungi that are different from the cordyceps and the Ophiocordyceps and the Metarhizium, those types of entomopathogens that we often find on summited insects. These are earlier diverging fungi, and really interesting how they change the behavior of their hosts. I won't go into the behavioral modification tonight, but I will talk a little bit about some of the molecular and morphological work we've done on some of these fungi. I've been working in the Hypocoreales for a long time. I did my master's at the University of Maine working on beach bark disease, which involves two closely related Neonectria species. But I also continue to work on Fusarium and Neonectria and Coronectria, Canoidea crella. And these are both plant and insect pathogens. And Fusarium is also an insect mutualist. I've taken the liberty to highlight some of the groups that I specialize in red here on the tree, just so you can kind of see where they kind of fall in the fungal tree of life. I'll touch base just one slide or so on some work I'm doing on Botrytis feriales, which include Diplodia and some other fungi, as well as some maple plant pathogens that I've been involved with recently. And that's pretty exciting work because it involves a fungus that's very charismatic that I'd never worked with before. So I want to start off by talking about a chance encounter I had with entomologist and millipede taxonomist, Dr. Paul Merrick, a colleague and friend of mine down at Virginia Tech in 2013. I was a postdoc there, and he was giving a talk, and he finished with a slide showing Brachycybe, which that's the genus of millipedes you're seeing here. And he said, oh, and by the way, there's a whole group of millipedes that obligately feed on fungi, and no one person has ever studied their fungal communities or their fungal diversity. And I was at the time working on ambrosia beetle fungi. These are fungus farming beetles. And of course, this just hit me square between the eyes. And I thought, oh man, I really want an opportunity to work on these millipedes. No one's ever studied them. That's like ripe for discovery. And that's really, I ran down the hall after his talk and I said, listen, we need to talk. I had already accepted the position at WVU, but sure enough, these millipedes, which I'm going to talk more about over the next, the next many slides, is a group of millipedes that occur here in the East, including West Virginia. So it was a real opportunity to kind of dig into what was going on with these millipedes and the fungi they consume. Just a quick millipede classification slide. If you don't know much about millipedes, these are segmented arthropods with 2 leg pairs per segment. They differ from millipedes who have 1 leg pair per segment. Really interesting is that millipedes are some of the earliest air-breathing animals, dating back some 420 million years. There's an estimated 24 to 80,000 species. And like a lot of small invertebrates, they're highly understudied and underdescribed. We know they live underground in wet, rotting substrate, and most of them, are detritivores. They serve the same purpose that earthworms serve. They're turning over material. They're breaking down material. In fact, they could be displaced by invasive earthworms. So they serve a really important role as native species in our habitats here, particularly in the central Appalachian region. They can physically break up substrate. They can tunnel. They can split apart rotting logs. They can shred leaves. But I'm really going to focus on the Colognatha, which is a subclass or subclass of millipedes that broke away from their detritus or leaf-feeding relatives about 250 to 300 million years ago. And they said, you know what? Eating leaf litter is boring. We want to feed exclusively on fungi. So when you ask, if you're ever asked who were the first to feed on fungi, it was the fungus-feeding millipedes, and it was almost 300 million years ago. So a little background on these Brachycybe millipedes that I studied. This is several pictures of them. They're pink. They're found underneath logs. They're quite interesting. There's 8 described species, 5 of which are native to the US. The other ones occur in kind of East Asia. We see some in Japan and in South Korea and other places. And certainly there's more that have been not discovered or described yet. Brachycybe lecontii is the one I'll focus on, and that's found in 13 states. And this is just a poll from iNaturalist that shows kind of where they've been reported on iNaturalist. So you can see we have populations in California and then populations in the East. Some real interesting social and feeding behaviors. First of all, they feed in these multi-generational groups. If you look at letter B here, you can kind of see them atop a fungus feeding. There's reproductive division of labor. This is one of the few animals where there's an example of exclusive paternal care. Males guard the eggs. The females lay the eggs on the male's legs, and the males guard them until they hatch. They also have these pinwheel-like colonies they form. We see them feeding on rotting logs and leaving behind divots. And many of them we observe are feeding on flevioid polyporales or polypores. But really, the literature on these millipedes is one sentence in a paper from 1974 that said, Brachycybie feed on Peniophora. And as probably a lot of you know, that's a Rushalales member. But really, all the literature on fungus feeding millipedes was boiled down to a single sentence in a publication from the 70s. And that was really motivating for us to figure out what was going on. We know just by looking at iNaturalist at these millipedes that they're feeding on a diverse number of fungi. I mean, we see Urpex, we see other fungi there, but it's clear that it's not just one fungus. So we have the visual proof that they're feeding on multiple fungi and could be generalist, but maybe not. But there seems to be a pattern where they're feeding on members of the polyporales. Whether or not that holds to be true, you'll have to just stay captivated and keep watching. But really, our research questions were, do Brachycybie millipedes maintain a cultural fungal symbiont on which they rear their young? And are they mutualistic fungi, white rot polypores, given that they can be typically found feeding atop these on the underside of logs? So we sampled 313 millipedes from 60 colonies spread out across 20 locations in 7 states. And we also sampled all the logs they were on because we wanted to know what the wood host was. And we dried those out, sanded them, and then I took forestry as an undergrad, so I used Wood ID to identify those. And we counted some 5,154 CFUs. That stands for colony forming units. And if you look at these plates, you can see each colony represents a CFU. So we had 5,154 colony forming units across 313 millipedes. And that resulted in 1,310 isolates that we retained for basically the ITS barcode sequencing. A lot of these, when they're growing on plates, they don't look like what they look like when they're fruiting on wood. So identifying them, at least at first, when you start a project like this and you don't know what fungi you're going to encounter, could be a little daunting. But certain patterns start to emerge, and certain fungi, old players, show up, and you recognize some of those things. We basically found fungi spanning 4 fungal phyla, 40 fungal orders, and 176 genera from one species of fungus feeding millipede across this geographic space. How do you make sense of that? Well, it's hard to do, but if you have all this metadata, like what wood host they were on, what clade the millipede was in, so we have molecular data for the millipedes, we have molecular data for the fungal component, we can basically establish a network and try to basically build their food web or whatever. It doesn't necessarily mean it's serving a food role or a nutritional role. It could have a defensive role or some other role that we can't really figure out yet. But this is one way to look at fungal diversity. This is similar to a network analysis you would do for social media. Sometimes you wonder, well, how did that guy from Southern Utah end up commenting on my grandma's picture she posted on Facebook? Well, it turns out we're all connected through other people to other people, and eventually, the degrees of separation keep increasing. But when you boil it down, you can find some core patterns and some core players. For example, this clade 3 of the millipedes was the most dominant genetic variant of Brachycybie lecontii. And what we found was there basically 12 core genera identified from 9 orders using culture-based approaches. This included 3 members excuse me, of the Mucora mycota, including Mortarella, Mucor, and Umbellapsis. I'm sure some of these are familiar, at least Mucor and other Mucora mycota that probably grow on your compost piles and things like that. We also found some dematiaceous or black yeasts, including Phylophora, Ramichloridium, and Fonseca. including a new species of Fonseca. We found some entomopathogenic hypochondriales, including verticillium and sectorum. One polyporales, a little disappointing given how many polypores we saw them on. That was Phanerochaete. And then 4 other fungi, Trichoderma, Penicillium, Cosmospora, and Silaria. So, you know, we get to understand who the core players are, but their roles remain really unclear. We just know that they serve some important role in that system. So how do we take it a little further? Well, we can collect millipedes. We can grow fungi. This is a culture-based study. We can grow cultures and, you know, and as pure cultures, and then put the millipedes atop them and ask and answer the question, what do the millipedes do in the presence of only fungus X or fungus Y? So we challenged them against 30 different isolates for 168 hours, including 7 of the core community genera. And we placed them on these fully colonized Petri plates. A majority of the treatments did not cause mortality. That's not a huge surprise because we're thinking we're identifying the core fungal food web, that these serve some form of nutritional role. So they shouldn't have a detrimental effect. They should have a beneficial effect. What was really interesting is that 6 polyporales and 1 entomopathogenic hypochondriales, that was Metarhizium, caused significant mortality. Now here we are positing that the polyporales serve some important role, yet when we put them on pure cultures of these fungi, it kills them almost immediately. And that was really surprising. And that's kind of what this chart is showing over here, the ones that actually caused mortality. And this is a survivorship curve here, where we can kind of trace how much time it takes for millipedes to die. So that was surprising to us. But we also recognize that when fungi are in a growth phase versus a fruiting phase, well, they're going to produce different metabolites or different small molecule chemicals. Because when you're navigating a new substrate versus fruiting atop it, you're going to have different levels of secreted small molecules, and that could affect your interaction with these millipedes in pure culture. We did uncover 2 new fungi that we're moving forward with describing. One was a close relative of this Mortarella ambigua. Actually, Mortarella has just been broken up into 13 new genera, and this is now Actinomortarella. But we basically found 27 isolates from 3 states. So this was a real common fungus that was real interesting. It produced these really interesting lipid-rich structures in the agar, and we're trying to ascertain the role of those structures, but we still don't know as of yet what that is. We found a new genus closely related to Apophysamyces. Now that's a really important human pathogen, opportunistic human pathogen. After the Joplin, Missouri tornado back in 2011, about 13 people got Apophysamyces infection, and almost 60% of them died. It's a very aggressive pathogen. We found this one in Oklahoma, so it could be a putative new human pathogen. I didn't inoculate myself and try it, but given the high mortality rate, I think that would be a fool's errand to do. And we also found 5 other new fungal genera or species, but we need more molecular data to resolve them. They may have been like only a single isolate, and you know, maybe that's a group that's taxonomically understudied, so there's not real clear species boundaries. But what about unculturable fungi? Because, you know, remember, we can only culture a small amount of the fungal community, or the bacterial community, or the, you know, whatever organism or microbe you're looking at. We can only capture a little using culture-based techniques. So as kind of a follow-up to this, we wanted to identify the fungi associated with using culture-independent methods. That is just DNA Illumina sequencing, so DNA barcoding. So basically, we went back to 2 of our sites where we had sampled for the culture-dependent study, and we added a 3rd site, and we collected 20 millipedes from each site, and wanted to test whether or not we'd find those same core community in the culture-independent study. And we used a Illumina sequencing, and we had some mock communities, and so we could basically tell if we were finding the same fungi from the culturable community and the unculturable community. There's a lot of details in that I won't go into too much, but really we were just hoping to capture a wider spread of the present diversity in these communities. And sure enough, just to kind of recap and my student Angie Macias's work in 2019, which was the culture-based study, we found 176 genera of fungi in 40 orders. That was 300 millipedes sampled from 20 sites in 7 states. So this work, which we're getting ready to submit, revealed 621 genera of fungi and 100 orders can be sequenced from just 52 millipedes from 3 sites in 3 states. So this is all the same species, this is all Brachycybelecantii. And when we're able to do direct comparisons, we can say 38 fungal orders were shared between the 2 studies, 2 were unique to the culture-based study, and 61 were unique to the DNA-based study. It's not a surprise that we got way more diversity using DNA alone because, again, some fungi are unculturable. Many fungi are rare or low incidence. So capturing one spore in a serial dilution plating, or if you get a fast-growing mucor or something that could outgrow it, then you might not be able to capture the slow-growing fungus using culture-based techniques. And you can kind of see the diversity here. You can see the Dothidiomycetes, the Sordarioomycetes, and the Leoshiomycetes represent, almost half of all the millipedes core fungal community. We had to do it at class level because when you do it at order level, that's 100 orders. Just color coding that, it's chaos. And I'll just to kind of reiterate that it's chaos. This is just the diversity of hypochreales across the millipedes. I want to point out that basically the y-axis are unique hypochrealian fungi. The x-axis are individual millipedes that were sampled. So you can kind of get a fingerprint of who's present. You can see, for example, trichoderma, which is a really common environmental fungus that grows on wood, is common pretty much in all the millipedes. Not a surprise there. And it's an ideal mycoparasite. So that's pretty interesting. Polyporales was one we were interested in, although we saw some mortality based on the culture-based bioassays. You can see that there's a lot of Phlebia and Phlebia-like polyporales here. We can see a number of things that I'm sure a lot of you recognize, Burjicandra and Flavodon and some other interesting polyporales that we see them on and appear to be pretty abundant, but quite diverse across the millipedes. Another interesting fungus that we found present in every single millipede, or all but one, was this Oidiodendron fungus. So this is a member of the Ongenales, and these are really good at consuming and breaking down keratin, mostly found in the soil. We did a plating assay and found that they didn't harm the millipedes. So, and this is just basically a tree we put together where we pulled sequences from the culture-based study that we found Oidiodendron in the culture-based approach. And they're labeled with BC designations. And then the OTUs are from the culture independent work. So you can see it kind of groups, it clusters together in these. They're not identical. I mean, some of these are, but there are some differences in those DNA sequences, but clearly form a clade and might have some important role. But what that role is, we don't know. All we could say right now is that it doesn't kill the millipedes when they are put on it for 7 or 14 days. So what are the big takeaways here? Well, Brachycybelecantii associates with a diverse group of fungi spanning some 621 genera and 9 fungal phyla. Many of the conserved fungal community members from the Macias et al., like my students' 2019 culture-based approach, hold up, has high incidence fungi in the system. The one huge caveat here is that ITS is not ideal for early diverging fungi. So we didn't find a lot of the Zolpagomycota. We found some, but we didn't find a lot of them. We didn't find chytrids. We didn't find those. And we know that the ITS primers that we use standard for Illumina sequencing miss a lot of that early diverging diversity. So as a follow-up to this, we're gonna go back through with the 28S or the LSU primer set and see what kind of fungal community structure we can get using those approaches. But what about the fungal communities of the 97% of other described fungus-feeding millipedes in the United States and elsewhere? You can go on iNaturalist and look at fungivorous millipedes and see that they're widespread across the US and they're widespread across the world. Well, we got a National Geographic grant in 2020 to study the diversity of almost 70% of the US species. And this took us on 2 major expeditions in 2021 for first to the Southeast United States as a collection spring break where we traveled 3,500 miles and collected 84 millipedes from 5 species. And then in the Western United States in late November, early December, we traveled 2,500 miles from Phoenix, Arizona, all the way up to Arcata, California, into the Sierras, and then back down to San Francisco. And then we sampled another 12 species from 8 genera. We've collected some other ones in between with small trips, and that work is ongoing. But we're going to compare the fungal communities of B. lecontii with these 20 plus millipede genera we collected for both DNA-based and culture-based approaches. So we're really excited about that. Should be seeing that work in the next year plus coming up. So, in reality, we're just beginning to scratch the surface on these enigmatic animals, despite them being the first terrestrialized land animals and obligate fungivores. Millipedes are generalists, but there appear to be some preferred fungi, including some polyporales, some black yeast, so some entomopathogenic hypocreales. There's still some contradictions that we have to reconcile, but we're just getting started on this. And really our National Geographic supported project hopefully will provide more clarity on how certain fungal groups are conserved across larger swaths of millipede biodiversity. But that kind of, to make it abrupt transition, I work on entomopathogenic fungi and fungus arthropod interactions, but one of my other loves is on the Nectriaceae, looking at some of these canker fungi. And some of you may be familiar with beech bark disease, which is it's a disease complex that involves both an exotic scale insect, Cryptococcus phagosuga, and 2 closely related Neonectria species, Neonectria faginata and Neonectria datisma. And here we could see a scale-infested beech tree, and here we could see a beech tree following scale infestation that is just heavy fruiting with Neonectria. There's a bit of a difference between the 2 species that cause beech bark disease. Neonectria faginata is only known from beech in North America following scale infestations, and has not been found in the absence of beech scale or prior to beech scale arrival. And that stands in contrast with Neonectria datisma, which is known from the US and causes perennial target cankers on a number of hosts, including birch, maple, walnut, sweet gum, mountain ash, a number of interesting hosts. And an interesting study by Houston in West Virginia between the 1980s and the 1990s showed that basically Neonectria datisma, which has these reservoirs on other hosts, initiates beech bark disease, and then over time is supplanted by a more aggressive Neonectria faginata. And that's a really interesting trend we'll come back to. But if you go into an aftermath forest where beech bark disease has been present a long time, you seldom find Neonectria datisma. You always and often find Neonectria faginata. Beech bark disease was discovered at Gone near Nam, an area that I love and spend a lot of time on the Mon National Forest down near Durban. And this was discovered back in 1981. Beech bark disease now reported from at least 18 West Virginia counties. You can see this map here in red. And in 2017, we detected it. I see my dots shifted a little bit when I moved the map, so I apologize for that. But we detected it in the botanical gardens in Mon County in 2017. But areas kind of to the west of I-79 are pretty much beech bark disease free, where you get these North Bend State Park and areas out towards Parkersburg have beautiful large diameter old growth beech. So one of the things that I wanted to do is I worked on beech bark disease while I was in Maine, and because we found beech bark disease so close to the university, I really wanted to resolve host and species boundaries for a number of these parathecial producing nectriaceous fungi across the central Appalachian Mountains. The reason being is that when you find these parathecia, which are much smaller fruiting bodies than a mushroom, think about nerds candy. If you were to glue a bunch of red nerds to a tree, that's basically the size of these fruiting bodies. So it's really hard to tell species or get species level resolution in the field. So what I wanted to do is go out and with my student and sample as many parathecia on these trees as we can find, and then identify who's where, what their hosts are, and then maybe test a little bit more about their pathogenicity and host specificity. So at each site, we went out and we sampled beech trees because we were trying to find other players in the beech bark disease pathosystem. We sampled target cankers on birch and any host we could find. And we took these bark plugs and then we were able to isolate out. Basically, we would squash the parathecia on a watch glass or the parathecium and then basically do a serial dilution plating. And as these individual ascospore colonies came up, you would pick individual colonies and then you'd have single ascospore colonies. And then you can not only identify them using molecular approaches or spore morphology, you could also do mating type assays because you basically have a single colony of a single mating type. So we then went on to characterize them molecularly and figure out who was all there. And this is just some of the parathecia and the corresponding culture diversity we saw across various hosts. And this spanned both angiosperms, hardwoods, and conifers. We were mostly focused on hardwood hosts, but it became clear as we were kind of in that area at Gonnier Knob and Spruce Knob, right, where the conifer forest meet the hardwood forest, that there was some interesting dynamics there and it was worth pursuing some of the conifer hosts as well. And this is a big table, so I'm going to summarize it in a better way here. But basically, we sampled 1,257 parathecia, and we sampled them across 13 sites, including 920 from beech and 337 from 11 different hosts. You can see the hosts include a number of maples, balsam fir, striped maple, black mountain maple, birches, black birch, yellow birch, beech, mountain holly, tulip poplar, red spruce, mountain ash, basswood. 53 of the sampled parathecia from beech yielded Neonectria, whereas the rest of them, that's over 850 of them, produced Neonectria faginata. 26 yielded this Clonostax rosea, and then 841 yielded, Neonectria faginata. What was really interesting is that we were able to not only confirm Neonectria datisma on a number of hosts, which in the literature said they were hosts, but had never been confirmed using morphology or DNA. We found 3 previously unreported species, and that included mountain ash, mountain holly, and mountain maple. So a lot of these mountain species harbor Neonectria datisma and have target cankers. We also found a Neonectria neomacrospora, which is a closely related species, and I'll show you where that falls out phylogenetically here in a second. So in addition to kind of surveying who's who, we also did a multi-gene phylogeny, so we can figure out the relationships among these Neonectria species. So a couple things I'll point out here is that we found Neonectria and Coronectria species, the Coronectria being from conifers, the Neonectria being from, except for Neomacrospora, all the other Neonectria were from angiosperm hosts. We found Neonectria datisma from diverse hosts, but they formed a single lineage, so that makes sense. They're all the same fungus. What was really interesting is we were able to confirm that Neonectria magnolia is actually a distinct species from datisma. Previous literature stated that it was a synonym, but clearly this was first described from West Virginia in 1943. We were sampling from the same regions and found it both on Fraser magnolia and tulip poplar. So we're able to basically say, hey, this is its own species and it's sister to these other beech bark disease pathogens. So that was really exciting as well. So like I said, Neonectria magnolia was a legit species. And actually, as we move forward with this, we're suspecting it might actually either be 2 distinct species or there was a host jump here. And follow me just briefly here. Here in black, we see isolates from Fraser magnolia. And then down here, we see basically a divergence, sequence divergence that results in this kind of branch and this group from tulip poplar. Now, not only are they different based on DNA, but what's really interesting is that only isolates from magnolia produce both macro and microconidia. So the macroconidia are missing from the tulip poplar. So as if to say the host jump caused it to change somehow and lost its ability to produce macroconidia. So it's pretty interesting. We're continuing to explore those relationships and try to figure all that out. One of the other interesting things that came out of this work is we found a new player in the beech bark disease system. We found a Fusarium bambinda species complex members that was present in a lot of the trees, but lacked sexual fruiting bodies. It was sporulating atop and alongside the parathecia. You can kind of see the white here. See the macroconidia that are characteristic of Fusarium. We looked in sites without beech bark disease and found that basically it wasn't present. So it's definitely associated with beech bark disease, and we think it may be colonizing the scale populations on the way out. So it's acting as an opportunistic entomopathogen. But we're trying to figure out more about that. Of course, as a pathologist, I care about pathogenicity, and these kind of aligned with what we had previously thought. For example, Datisma was a strong performer on a number of hosts on which we can find it. But really on tulip poplar, we see the Neonectria magnolia indicated here in the tall gray bar outperform Datisma, and that makes sense because it seems to be a specialized pathogen on members of the Magnoliaceae, or the Magnolia family, which includes Fraser magnolia and tulip poplar. Some pretty interesting results, and we continue to ask more questions from these. But clearly there's a lot of diversity of Nectariaceae across the central Appalachian region. And we're finding a number of them, including 9 species from 11 hosts. Vaginata continues to dominate the beech bark disease pathosystem. And we show that these tulip poplar and magnolia strains of Neonectria magnolia are kind of divergent, and we want to explore that a little bit more. Some recent papers that I was involved with kind of took a DNA-based approach where we just basically amplify DNA from bark samples with beech bark disease and showed that Datisma is actually more present than previously thought. It's just not fruiting as readily as Neonectria vaginata, so that could explain why we're not seeing it. They're both heterophallic, meaning they have 2 separate mating types and need 2 mating types present to form fruiting bodies. So there's not a difference in phallism, but there is a difference in fruiting success. Probably is either substrate driven or temperature driven. Another interesting result that came out of that follow-up work is that we showed this Fusarium bambinda, as indicated in green in these pie charts, is really common across the beech bark disease pathosystem, and an important player, presumably, but yet we still have to further explore its role, but it's there all the time, everywhere you look. One final update I'll say is that we recently uncovered a new Neonectria species on old growth maple down in Pocahontas County. One other strain of this had been deposited previously from the Horseheads region of New York in the southern tier. So we're trying to understand the role of this fungus potentially in maple decline. But pathogenicity tests are underway, so we hope to have some more information on that. But it clearly forms a unique clade in the Neonectria punacea species complex, which is, pretty much grouped in there with vaginata and coccinia and magnolia. So these are all closely related, and they're all serving kind of unique roles on different hosts in the same environments. So let me transition again back to insects. In 2016, brood 5 periodical cicadas emerged in Morgantown. I was fortunate enough to be here during that time, and if you've never seen a cicada emergence, it's quite something else. I'm going to pivot back to a powder mill announcement from earlier because we discovered something really remarkable at powder mill, and I'm going to show you in just a few slides. As you may or may not know, cicadas, like a lot of other insects, have a number of parasitic fungi that attack them, whether it be Ophiocordyceps or Buvaria or Metarhizium. I believe this is John's photograph. I can't see the bottom of it, but I did put a citation there. But of course, we're interested in Mycospora, this early diverging member of the Zoopegamycota. If you've never heard of Mycospora, this is a member of the Entomopterales. There's at least 13 obligate sexually transmissible fungal pathogens that infect 21 cicada species worldwide. This was a genus that was established by Peck in 1879 with the description of Mycospora cicadina from periodical cicada from New York. But really a mycological oddity up until Richard Soper stepped in the 70s and basically wrote the first monograph on Mycospora and did some really remarkable work in several papers on this important cicada entomopathogen. We were lucky that during 2016 that this happened right on campus, and we were able to acquire a lot of infected cicadas. Periodical cicadas is not just one species; it's could be up to 3 species co-occurring. We collected males and females, all with these Mesospora plugs. You can see the fungal plug emerging from the abdomen. It's replaced the abdomen, kind of takes on the shape of the abdomen. We were lucky also that we have a number of collaborators that had or got us access to Mesospora platypedia, Mesospora levospora, Mesospora disroprocta. So we're able to get these from cicada scientists and other people that had kind of had them as bycatch. So we were excited to be able to basically do extract DNA. And just to kind of show you how this looks across different cicadas, we have the periodical cicada with Mesospora cicadina. Then we have annual cicadas infected with Mesospora levospora. This is Oconagana. This is platypedia infected with Mesospora platypedia. This is disroprocta infected with Mesospora disroprocta, and Pterygotus cicadas infected by Mesospora pterygotus. And you can see they both have these conidia as indicated in the top, and then these thick-walled resting spores as seen down the bottom. And they don't have both spore types co-occurring. The early infected ones develop these conidial plugs, which you can see here in the pictures, and then subsequent interactions between healthy and infected cicadas result in the resting spore production. So because these are mycological oddities, we really had to go back to the literature and say, well, in order to characterize these, we have to use morphology at first to basically link with the historic literature. And we showed that really only with width do you see this differentiation of these species. If you look at something like spore length or resting spore diameter, you don't really see impact. But we looked at number of nuclei, we looked at the ornamentation of the epispore, a number of features. The really cool thing is we generated the first DNA sequence data for this genus, and we're able to do a phylogenetic analysis to understand the relationships between these closely related Mesospora species. We see, for example, that Mesospora cicadina is sister to Mesospora disroprocta. And another interesting result that kind of knocked us down is that Mesospora platypedia and levospora, which infect different cicada genera in different geographic regions, are actually the same fungus. And we showed this conclusively with multi-gene phylogenetic analysis. And even the spore morphology aligned. So that was really interesting. And we officially synonymized those in our 2020 paper. We're continuing to do more of that work because we continue to get more cicada specimens. And it's actually through places like iNaturalist. People are watching iNaturalist for us and saying, hey, do you know someone just uploaded a picture of a Mesospora infected key cicada? Here's an example. And we were able to get this cicada. The guy didn't collect it, but he was still there. He went back 6 hours later, collected us 4 specimens, and we generated the first DNA sequences ever for this cicada host. And that was really exciting to leverage the power of iNaturalist to get specimens. One of the really other cool projects that I kind of alluded to was we resequenced the genome of Mesospora cicadina. And this was from a population we collected at Powder Mill Nature Reserve back in 2019. And it turns out that Mesospora cicadina is the largest fungal genome ever. Now, fungal genomes on average for ascomycotin fungi and other fungi, we're talking 30 to 50 megabases, million base pairs. Rust are a little more complicated, 100 million base pairs, 300 million. This is 1.5 billion base pairs. So this is 20, 30 times bigger than other fungal genomes. And it's a real perplexity because it's mostly filled with garbage. We call this stuff transposable elements. So we're trying to understand the evolution that led to basically the largest genome ever. We started studying this because we had it right accessible to us outside our front door. And it turns out that it's a really important discovery for kingdom fungi because it represents these hyperinflated genomes with really unknown function. And we can learn more about the wacky corners of the fungal tree of life by looking at Mesospora. Some other projects that I'll just quickly mention, this is like one slide apiece. is that I've been really interested in diplodia canker on red and white oak. In recent years, there's been this rapid white oak decline of white oak species members, including white oak, chestnut oak. And there's also been these bleeding cankers on red oak. And it started in 2016 or 2015. I started to investigate this with members of the Forest Service and some students. We basically found two diplodia species, Diplodia corticula and Diplodia corsivera. These are generally this canker disease called bot canker, and involves Botryosphera, but other closely allied fungi like Diplodia, and we basically found both present, and you can kind of see the diversity of isolates we found from both red oak and white oak. And we found this Diplodia sapinea as well as Botryosphera dothidia. You can kind of see the morphological diversity here and the diversity of symptoms that we see on the trees that are impacted by this as well. One of the other recent projects that I've been involved with is looking at maple decline associated fungi in Maryland, Pennsylvania, and West Virginia, going around to different sugar bushes and trying to understand the contemporary threats that these maple stands face in terms of pathogens and pests. Of course, we get to work on things like northern tooth fungus, which is such a remarkable fungus, on Utapella canker, which is a real enigma. Other things, getting to work with brittle cinder and mossy cap polypore, and maybe some new putative pathogens like Stegonosporium and this Neonectria panacea species complex member that we found impacting large old growth maple. One of the more interesting findings here, and I'll kind of leave it at this last bullet point, is that these Utapella cankers, as we come to, that cause these kind of large swellings, we've not yet isolated Utapella parasitic out of them. It appears to be a complex of other Xylariales, including Xylaria, Pneumonia, and Deldinia, and other members that may be contributing or causing similar symptoms, but it doesn't appear to be Utapella parasitica. There could be displacement of Utapella parasitica, but it could just be that these things for a long time are called Utapella canker, and they're not really Utapella. They're just more like Xylariales canker. One other kind of final project I've been working on is looking at these Polyporales mutualist of ambrosia beetles here in West Virginia. We found this Flavidon ambrosius found associated with this Ambrosiophyllus atreus ambrosia beetle. And this is a white rot polypore. We did a number of bioassays, including tannic acid tests and wood degradation assays to understand their role. But this was a discovery we made in the same woods that we were studying cicadas. So, everywhere we kind of look and spend a little time digging in, we can find some interesting things. So I know I've taken up a lot of time, so I'll leave it at that. But I'm happy to take any questions now. Thanks.
Yeah, wow. Thank you so much, Matt. That was incredible. Yeah, I do have a question. I didn't see anyone else put questions in the chat, but I put one in there. The target canker that you see on red maple, is that Neonectria dotissima?
Yeah.
Okay.
So we've sampled red maple before. It's we've never seen vaginata or magnolia on that. There could be other uncharacterized Neonectria or Neonectria-like fungi that could impact red maple, but definitely Neonectria dotissima occurs on red maple.
Okay. Yeah, that just seems to be a really distinct one that I see in almost any forest that has red maple populations. It like very clearly forms that target pattern. Richard and I both posted some observations in the chat if you wanted to see our pictures.
Yeah, there's that target pattern, but I was just wondering, I've never seen any parathechia or anything on that.
Yeah.
Like I was wondering, how is it just like so?
Oh, that the bark malformation that you posted on the apple canker there.
Yeah.
So I've seen that. That pattern I see mostly on like buckeyes and horse chestnuts. I see a lot on that actually. That's different. I don't know what causes that.
Okay.
Could be a genetic mutation. But I will say this, you know, coming back to just because you don't see parathecia or the red kind of grape-like fruiting bodies doesn't mean that it's not neonectria. There are a number of hosts, and sweet gum is a great example. We have a plantation of sweet gum at the university forest, and there's no less than 100 cankers on maybe 40 trees, and not a single parathecium occurs on any of those. Yet when we microsample the margins, we can get neonectria datisma out. It's just, it's not a suitable substrate for fruiting, and it might be some inhibitory effect of the sweet gum sap or something, or some kind of nutritional deficiency that exists. We're trying to better understand what enhances parathecial production so we can try to understand the discrepancies between culture dependent and culture independent results with regard to neonectria datisma. It's clear that datisma is on a lot more beech than culturing alone will reveal, but why it fruits less, even though it's got the same kind of phallism, you know, as neonectria vaginata, it remains kind of a mystery, but that's what keeps me going, I guess.
Yeah. Well, thank you. Kara also says she has a question.
Hey, yeah.
Hi.
So I came upon something. I believe it was now forgive my butchering of Latin here. Stylobrica, Stellobocria. Does that sound familiar? It's in the Nectriaceae family.
Okay.
I didn't know it, like, I don't know if it was even on a conifer or an oak because they were both there, but I'm presuming an oak because it was actually on a branch that my son found hanging. And
Yeah.
I haven't seen any other, like anything else like that on INET for really for the US. So I didn't know if that was something like that you guys find, you just haven't posted, or if that's a common thing.
Yeah, so one thing I'll say is that we work mostly with stem cankers because you can easily approach the stem and sample it and look at the stem. You have to rely on wind or a bucket truck to really look at what's happening in the crowns and the small diameter branches. But definitely there's a whole unexplored world.
Oh, so this would be like an upper part. That's probably why we found it on a branch that fell rather
That's right.
than on.
Yeah. So I think there's a lot happening in the upper crowns of trees, of hardwood trees in the central Appalachian region that we've yet to even because I do find some occasionally that are blown out and I'm like, there's parathecia, you know, and I'm like, I don't have time to look at that, but I you know, it's probably something interesting. But I have a new student coming on. Thankfully, she's got an NSF GRFP, so we're going to be further exploring this. So we may look at kind of those spatial dynamics in the canopy versus.
Yeah, that's interesting.
Yeah,
because that's you know, that's a big part of, you know, the ecosystem is the tops of the trees and themselves and all the different effects that can be had by having something harmed up there or some kind of parasitization up there.
Yeah.
So.
The other thing I'll mention that might help clarify it too is that the neonectria fungi, for the most part, are pretty weak pathogens. They usually enter through branch stubs, or you know, as trees naturally shed their branches, those tissues become moribund and kind of they don't have a defense response. So that's how those pathogens are able to gain access to the trees, because they're like, you have a dead limb, I'll enter there. And oftentimes when you find a nectria or a neonectria canker, you find a branch stub in the middle because they took advantage of a moribund tissue to get in. Now, once fungal succession takes place, and that's happening all the time, they're quickly hyperparasitized or supplanted by other white rot fungi or other fungi. So the evidence that they existed are kind of wiped clean. So as branches fall, you don't see parathecia or the remnants of parathecia because fungus X, Y, Z, AA, AB, AC, AD moved in and basically took advantage of that substrate.
Yeah, that makes a lot of sense. All right, well, thank you.
Yeah, that's fascinating.
I see that neonectria posted. Yeah, that's a classic beech bark disease canker there.
Okay, so And that just from like a photo like that, would you be able to tell the difference between the two species you find most commonly on beach, or is it really you got to look at the micro features?
No, well, you can look at ascospore length and you can look at colony morphology, but generally if you show me the stand and if you tell me there's 10 trees and they've had it, 10 years, I'll tell you that it's a 95% chance it's vaginata.
Yeah.
And I'd be shocked to find otherwise. But one thing is that it's always good to check, because as much as mycologists, we specialize things and feel really confident. Sometimes something shakes up our worldview, and like I think periodically checking in makes you discover new things that you weren't expecting, and that's actually the fun part.
Yeah. Awesome.
Mommy, see this guy?
Sorry, Kara, I muted you.
Did anybody mention anything about the Twinkie fungus in the chat?
I didn't see anything about it.
I didn't see you put a slide up for that. Like I followed that on Twitter or whatever.
Yeah.
Not that I'm on Twitter except for fungus. But
yeah, for those
that
of
was
you
really
that don't,
cool.
Yeah, thank you. For those of you that don't know, we had a moldy Twinkie that we got from this guy Colin Purrington, and it was a big piece on NPR about it. Now Greenfield Voice, and we were on Shortwave podcast. I also did something a couple years ago on Easter where I inoculated peeps with different fungi, and that was covered in the New York Times. So I'm constantly trying to engage the public and kind of outreach. The reality is this: if you want to get people interested about fungi, don't show them a DNA sequence or a Petri plate, because that's not something they have at their house. But sure as hell, they have a Twinkie and they have a peep, Easter peep. So if you can kind of shake up their world by meeting them where they're at and showing them what fungi can do to kind of unusual substrates, you get them thinking about fungal ecology and, wow, what other roles do fungi serve in the environment? So that's really kind of been my mission, is like get them hooked on some grotesque fungus mutilated peep and then kind of reel them in and teach them a little bit about fungal biology.
Yeah, that's a great tact. I love it.
Yeah, genius. Yeah.
Other questions?
Kara posted her INAT observation of the Stilbocrea.
Is that, let's see.
If you wanted to take a look.
Oh, yes, I've seen that before. I saw that on a Olympus tree, like back in 2009 in Dauphin County, Pennsylvania. And I don't think I have a DNA sequence for it, but I'm pretty sure that's what it was. Yeah, I don't see that one that often, but you're right. It's not like, for a guy that looks at sticks all the time, me, I don't see that one very often, but that's definitely a cool find.
Yeah, I've never seen anything like that. It almost looks like a slime.
I even got the spores on there.
Yeah, no, it's a beautiful shot. And the thing is, with regard to the Neonectria or the Nectriaceae, if you're on Twitter and you see these things, feel free to tag me, because I'm always interested in trying to find weird things and find more of the same things. So.
Yeah, so before your talk, you were saying, there's different species that you're interested in receiving samples of. Like if you had to say like your top 3 most wanted specimens, or like the top 3 things people are most likely to encounter, I don't know, is there any way we can help you out if we're out looking for stuff?
Well, I'm really interested in the Entomopterales, like the Mesospora and other type of Not so much, Entomoptera musci. I mean, we're doing a paper on that. That's not one I'm particularly interested in, but like there's a number of like the goldenrod soldier beetles get an Entomopterales. That's a really interesting one. So a lot of these interactions. I'm always interested in Ophiocordyceps on different weird hosts. I showed a picture early on of a cicada killer infected with an Ophiocordyceps. So we're always looking for specimens of that to characterize. But I'm interested right now in Clavicodon, the northern tooth fungus, which is really easy to spot on old growth maple. But it also occurs on more than sugar maple. It's been reported on beech. I've seen it on red maple. I'm interested definitely in the ones on beech and non-maple hosts because we're trying to figure out if it's all one species. I suspect it is. I have no reason to think otherwise, but like I don't know how much sequence data has been generated from these different hosts. So I would love to get some more northern tooth fungus, fresh fruiting body, or even GPS points of where one is on public land, or a DM on Twitter, an email.
Yeah, I found one on beach at Tom's Run here.
Well, it usually comes back several years in a row. So if you happen to get over there and you see it, during the peak season, and you want to get me a piece of fresh fruiting body in a Ziploc bag and put it in the fridge, and then we can kind of work out how we'll transfer it. But we can culture it quite well. We have several cultures now of northern tooth. We use like a hydrogen peroxide method for getting it to grow on culture media, and that grows pretty well. Just to kind of circle back to one of the things that was, maybe it won't work for this time, but I know that you do these inoculations, and we have cultures of so many edible fungi, including chicken of the woods, just some really Hericium. We have a lot of these things in culture that we'd be happy to share in the future.
Very cool. We just got another question in the chat if you have time for it.
Is there a future fungal pest control? Yeah, I mean, there's definitely work with entomopathogenic fungi for biological control of invasive insects. And there's a lot of money being thrown into it just because these things are hard to, sorry, my cat's in the background meowing like crazy. The problem is, well, if they're found naturally occurring here in the invasive environment, that's the best scenario because you're not introducing it from afar and you're not worried about non-target impacts because it's already out there. It's kind of like doing a field test for you. So yeah, there's a good future in biologicals just because, chemical controls will always be used, but integrated pest management is always better when you can combine cultural and biological and chemical controls in a way that minimizes the non-target effects or the environmental effects of chemical control. That's a good thing. So yeah, definitely. Oh, that's a nice one.
A request to see the cat.
I can look at iNaturalist posts all day. I don't have a personal iNaturalist account. I usually use Twitter as my main form of, not that they're the same, but our Millipedes account is for documenting fungus feeding millipedes across our for our National Geographic project. So we mostly have fungus feeding millipedes on there. But if you want to see some of those, we definitely have some in the area. There's some at Ohio Pile. There's some here in Morgantown. There's Petaserpes and some other species. So if you want to go looking for them, they're fun to look for.
Is there a time of year that they tend to be out? I know you said for your southeast.
Yeah, so not right now for Petaserpes, but summer through December, you can find them underneath logs. And it's usually really decayed, decorticated or bark-free logs along creeks. If they're covered in moss, all the better. you turn them over and you see these kind of, they're called slug millipedes for a reason. They look like little white slugs, but they are millipedes and they are fungivorous.
Wow.
And what they eat, well, I'll let you know. Stay tuned. Our Nat Geo project is looking at that.
Fascinating.
Oh, can you see my cat? No. She won't come into me. We have I have a Newfoundland dog, and she like now doesn't even like me anymore because I pay too much attention to the dog.
Awesome. Well, if there are no further questions, I think we'll stop our live stream here.
Okay.
And